In the beginning. Well, a little dramatic, but in the beginning, this may not be too far off from what the Earth looked like. It had just formed. It was probably still getting slammed by a bunch of rocky material from outer space. Chunks of the surface hadn't cooled off yet, and no liquid water. So, this is what our planet might have started out like. So, how would we go from this, which is an artist's illustration of what the early Earth and its atmosphere might have looked like, to this, which is what we see in the modern day? That kind of typical blue atmosphere that we see with lots of green on the planet, indicating that there's a lot of living things and a nice liquid ocean of water. Also, what this story involves is how did we go from this, which is a mix of random chemicals in the air, to this, which are these super complicated forms of chemicals that we see in living organisms today. It's quite a dramatic difference that we see. And how do we explain all of that? Well, it's with abiogenesis, or the abiotic origins of biotic creatures. So, how did life first arise on the planet? So there were, it wasn't something that happened in a day, in a year. The steps for life probably happened over hundreds of thousands, if not millions of years. Small things happening, some of the characteristics for life kind of accumulating in these pre-life or proto-life organisms. But some of these four steps here basically needed to happen before we could get anything that could lead eventually to modern life. So, molecular self replication is basically, can you make copies of yourself? Remember, DNA can be used to make copies of itself. It is able to be replicated. You can make RNA copies from RNA, though our cells don't tend to do that very much. And then the DNA that you have can allow you to make more copies of proteins. Then these things need to assemble on their own. They need to be able to put themselves together because before there were living things on the planet, there was nothing to sit there and say, I want these molecules to be pieced together with these other molecules. They needed to have autocatalysis. Remember, one of the key characteristics of organisms, of living things, is they have metabolism. They are able to break down some molecules, put together other molecules, and they're able to use energy. And that means they need to be able to control chemical reactions, which means they probably need something that acted like enzymes, if not actual enzymes. And they needed some sort of boundary. This could have been something like a cell membrane, it could have been something different, but it needed to be something to enclose any kind of protocells and separate them from their external environment. Because if you look at the internal contents of your cells, they're very different from the environment around them. If you want to look at single celled creatures living in the ocean, they're surrounded by a watery environment, but their insides have a much different chemical composition. It might have different internal conditions. For example, us as humans and other warm blooded organisms, we maintain different body temperatures from the external conditions. We call that maintaining homeostasis. And you cannot maintain homeostasis unless you have a boundary to separate you from the rest of the world. So, this had to start out somewhere, and where it likely started is with the composition of the early atmosphere. The early atmosphere would have been very different from today's atmosphere. While it would have had some water vapor in it, and nitrogen and CO2, those are all things that we find in our modern atmosphere, so that's not very different. The thing is, you may notice it's not going to have any oxygen. The early atmosphere had very little oxygen in it at all, if at all, because oxygen likes to react very, very easily. And so once any was out there in the environment, it would react with some other molecule and no more oxygen. You would also have some carbon monoxide, hydrogen, and sulfur compounds too. And so these are inorganic molecules. However, if you supply a bit of energy, It could be heat from just something very warm like a thermal vent, places where lava is coming out from underneath the Earth's surface and then cooling down. It could be supplied by lightning, which can provide with a lot of energy in a very small space. But anything that could provide enough energy to get these molecules to come together and form some of these very simple chemicals like methylformate, glycolaldehyde. These probably don't sound familiar, but they're very important building blocks that can be changed and used to make some of our more familiar biomolecules. And so, random occurrences with energy causing these chemicals to combine with each other could have created the precursors for modern life. And then these things had to eventually become organized into systems. Self organized, kind of spherical collections of lipids called micelles have been called a potential stepping stone towards the first living cells. A micelle is basically like a giant vacuole or vesicle. It's got an aqueous or watery solution on the inside, so you got H2O stuff on the inside. It can be surrounded by a watery solution, and then it will selectively allow certain materials into and out. And that's basically the kind of initial, most basic type of cell membrane you could have. Our cell membranes are much more complicated than just having a bunch of phospholipids put together, but the original ones may have been just simple phospholipid shells. Add to that some genetic material and the ability to not only replicate but build up some mutations along the way, and you've got what some people have called protocells, other people have called them progenotes, but they're basically the precursors to life. They weren't quite life yet, but they were getting there. So we have a few major hypotheses about how life may have arisen on the planet. The first one, starting in the beginning, the early 1900s, was independently proposed by two different scientists, and so it's named for both of them. The Oparin-Haldane hypothesis starts with the atmospheres or early atmospheric conditions. Little oxygen, slightly different chemical composition, and then if you supply some energy, which could be either lightning or ultraviolet radiation, because you can't actually have an ozone layer without oxygen. So low atmospheric oxygen means no ozone, which means no UV protection. Well, that could combine these small inorganic molecules into kind of some of the basic monomers, a few simple amino acids, maybe some basic nucleotides, and start to build up a collection of biomolecules. Then they hypothesized these could start aggregating or joining together, and they could form these structures that they called coacervates in an aqueous environment. So what is fairly obvious is that life probably arose in some sort of watery medium first. There's a couple of different hypotheses about where, but ultimately, we're bags of water with a bunch of stuff dissolved in it. And the early cells were probably very similar. We don't have any life on this planet that is independent of water. And so the formation of these coacervates would be like, okay, you have some proteins, you have some polysaccharides, let them mix, and they will form these coacervates, which can then build up. And then start to take in other organic compounds that were in the environment. Now, there were a couple of weaknesses with their hypothesis. One, there wasn't a lot of certainty about the exact composition of the early atmosphere. And then with this, it was hard to kind of see how these the smaller monomers would have become polymerized and joined up to form these larger chains of molecules. Over time, their hypothesis developed into heterotrophic theory, sometimes called the primeval soup hypothesis. Basically, it starts with, like I've been saying, the conditions of the atmosphere. So here we see a diagram showing you the relative amounts of different molecules in the atmosphere. So nitrogen was very heavily featured in the early atmosphere. CO2 looks like there was a lot more of it in the early atmosphere, which means it was probably pretty warm. There was some methane, there was ammonia, hydrogen, and carbon monoxide, and of course water vapor. And all of those were in a reducing atmosphere, which would allow them to form these chemical reactions once they got a source of energy. Then those could end up going into and building in water. They could get like rained out of the atmosphere basically and accumulated at some point in the water to form like a soup. And I say that with full quotation marks because it wasn't like a literal soup. But it's basically like a bunch of this stuff mixing in the water. And then these compounds could have gotten concentrated at certain spots. A couple of hypothetical locations are shorelines. I read some stuff quite a long time ago that was hypothesizing like clay banks of oceans, like clay beaches, could have been an important place for some of these micelles or kind of primitive cell membranes to develop. Hydrothermal vents are a very strong candidate for the places where life originated on Earth because it's got a lot of the necessary components kind of stuck in a small space. And it's also got organisms that perform chemosynthesis, not photosynthesis. So they break down chemicals to provide themselves with energy. And that is something that early life probably would have had to do because photosynthesis is a pretty complicated process and it would have taken it a while to develop. It's not something that would happen overnight. And then as these compounds accumulated, they would be able to join into these complex polymers and ultimately develop life, which would, happen in this watery soup. Some evidence that would support the Oparin-Haldane hypothesis was provided by a couple of researchers, and their names go in this experiment. So see, you do something special in science, you get to name it after yourself or your favorite teacher. The Miller Urey experiment was really pivotal in providing some evidence that the Oparin-Haldane hypothesis was possible. So what they did is they simulated some of these hypothetical conditions on early Earth. They put a mix of gases into a chamber that was the mix of gases that was hypothesized to be the early atmosphere. Then they had electrodes in there that would create sparks, which were there to simulate lightning. Then they had a condenser to simulate rain, because, water cycle would still have been in existence then. The cooled water would come down here. Then it would go to a chamber where it would be boiled, or at least heated enough that it would produce vapor, just like water evaporates in the water cycle today. And that water vapor would go up to join this atmosphere mix. And this cycle, this apparatus could be left running for days and days. And then every so often, they could take a sample and they could capture that sample, analyze it to see what kinds of molecules were found there. And the really neat thing is, from this completely independent, abiotic, no life involved process and apparatus, they were able to create some basic building blocks. They were able to form some amino acids, some basic parts of nucleotides, and it was at least proof that it is possible to produce some of these necessary ingredients for life using completely lifeless means. However, there were some problems. The predicted early atmosphere composition was probably off. However, further experiments, once scientists kind of updated their idea of what molecules were in the early atmosphere, other scientists would perform the Miller Urey experiment with those same chemicals and would find that yes, you could still create amino acids. Over time, one of the things that was a kind of issue with earlier hypotheses about the origins of life is that we've got this pesky central dogma. DNA contains the instructions for making RNA, which is used to make protein. Okay, but here's the thing. You need to have a molecule that's capable of passing on genetic information, and that also needs to be able to be created by something. If you don't have proteins made, well then what's going to be an enzyme? You need something capable of catalytic activity. DNA can't do that, and DNA is pretty useless on its own. It needs proteins to help it do everything. you'll notice in all of the lessons that we've done, DNA's only job is to sit there and be read. If you want to turn on transcription, you need some proteins, transcription factors. If you want to actually transcribe, you need proteins. You need DNA polymerase or RNA polymerase. If you want to wrap it up for storage, you need some histones. So DNA is it's a little diva-like. It doesn't really do anything besides its one task, and it gets all the attention. RNA, however, is a little different. RNA can actually form more complex shapes than DNA, and it already exists in a form in your cells that can catalyze some chemical reactions. We tend to gloss over the fact that ribosomes are made of RNA and protein, but a lot of it is RNA. A lot of the parts that are actually catalyzing the chemical reactions are RNA, so they help catalyze the formation of proteins. So you've had an enzymatic RNA complex in your cells this whole time that you learned about your freshman year in biology. Well, as research continued into, what smaller RNA molecules might be doing inside the body, scientists discovered ribozymes. Ribozymes are ribonucleic acid enzymes. So they shorten the name down to ribozymes. These are RNA molecules that can catalyze chemical reactions. So they act like enzymes. And another neat thing is they can actually self-replicate. So they can reproduce themselves. So there have been scientists in labs who've created ribozymes that are capable of reproducing themselves and, catalyzing chemical reactions. So these could have carried on genetic information and carried out chemical reactions, like what you see here. You see here is a ribozyme. It's got a target RNA that it binds to. Once it binds, it cleaves that RNA, and then it's left behind in its original form, just like an enzyme is left behind after a chemical reaction. So, based on this and other experiments, a hypothesis was developed called the Rna World Hypothesis, that early life was RNA-based instead of DNA-based. The reason why RNA would have been used initially is because of that functional flexibility, but unfortunately, RNA isn't as stable as DNA. It breaks down easier, and RNA is more prone to mutations than DNA. You don't have that whole semi-conservative replication where, you can recheck your replication to make sure you did everything correctly. You don't have that with RNA since it's single-stranded. And so DNA eventually came to replace RNA because it was more stable. But this must have happened very early on in the development of life. Why? Because after that, all organisms on the planet go from DNA to RNA to protein to pass information. The only exception are things that we can't even 100% call alive. Those are retroviruses. But all they do is they basically go, if you're a retrovirus, they squeeze in an RNA at the beginning. They still use DNA to RNA to protein as their method of producing protein. They just start with RNA and make a DNA copy. So anyway, that's RNA world. So here's kind of some of the steps that would have been necessary. So you have inorganic sources. These are probably small, like nucleotide precursors that come together to form RNA. That RNA can self-replicate, and it can also cause protein synthesis. So it can build some very simple proteins. If all of that gets shoved into a cell membrane, then you start having the precursors of a cell because you've got something self-contained, self-replicating, it can pass on traits, it can catalyze chemical reactions, and then eventually DNA could become the master template and then end up coding for proteins to catalyze all these chemical reactions. And as generations passed, mutations accumulated, this whole set of processes could have gotten more complex and more complex until we get to whatever ended up being the first life form. Probably an archaebacteria. There's also some hypotheses that there might have been some not quite alien, but some extraterrestrial origins for some of these organic compounds. And not saying that life like, it's not like saying aliens came here 4 billion years ago and planted the seeds of life on the planet. No, the fact is that some organic molecules can form in space. There's plenty of energy floating around, and there's in like gas clouds or in areas with asteroids and stuff. There's some of the basic building blocks of some of the basic inorganic molecules that can be used to make organic ones. And so chemical reactions happen, and then you can actually have some amino acids that are produced in space. And so some of those have been detected on meteorites that have fallen on Earth. And there's even some estimates that during certain periods of Earth's history where it was heavily bombarded, in fact, that's in the name, bombardment, basically hit with a crap ton of asteroids and comets and everything just kind of raining down onto the planet. If we were able to see the surface of the Earth at that point, probably would have looked as pockmarked as the moon, because a lot of things were hitting. And there is some speculation that there could have been millions of tons of organic molecules and materials falling on Earth every year from the late heavy bombardment. Of course, that would need to be proven with more evidence, and it's not to say that this was the ultimate origin of all organic material. It may have just been one possible source. So that is it. Next, we're going to do natural selection, and that may be split into a couple of videos. We shall have to see.